Hello and welcome back to another Teach Me in 10. Today we're joined by Phil Beckett, a Discovery Imaging Field Application Specialist at Cytiva. He will discuss the fundamentals of robust Western blot quantitation, from choosing the best normalization strategy to how modern fluorescent imaging enables the high-quality, reliable data required by top-tier journals.
Hello, and welcome to today's Teach Me in 10. Today we're going to talk about robust quantitation for Western blots. So why would we run a Western blot? So we want to basically confirm the presence of a protein in our sample. We'll do that by using an antibody. But then we can also run molecular markers as a secondary help to make sure that we're identifying the correct protein species. And then looking at the intensity of the band, we can kind of get some kind of quantitative measure on how much protein is actually present in that sample. So historically, people would have just submitted a protein blot to the journal and they would have published, and there would have been any questions. But the more we kind of work with Western blots, the more we know there's prone to errors in terms of loading or transfer. So we need some way to kind of help with normalization. But one approach is to normalize against the housekeeping protein. So hopefully this protein is constitutively expressed in your protein sample, and then we can take our protein of interest and divide by the housekeeping protein to do this normalization procedure. We can do that within a lane. In a chemiluminescent situation, you would basically use the antibody against your target protein of interest to start off with. And then normally you'd strip and reprobe to look for the housekeeping protein. And that housekeeping protein is usually in higher abundance, so there's less chance in that stripping and reprobing to lose that protein. And then again, we'll do that calculation where we'll divide our target protein by the housekeeping protein. So that will help to allow for this uneven sample loading or uneven transfer that might be going on. But another way to approach the situation without having to strip and reprobe is to use a fluorescent approach, where you can have two different secondary antibodies conjugated in two different species. So the primary could be raised in rabbit and mouse, and then you'd have a goat anti-rabbit with Cy3, and then a goat anti-mouse with Cy5. So in this situation, you don't have to strip and reprobe. You're seeing everything at the same time because you've got two different host species. So it makes it very easy to differentiate between the two targets. So here you can see on the left-hand panel, if we just did the quantitation without normalization, but now when we do the normalization, you can see we've tightened up the data. We can see that actually there's lower expression of these two proteins in lanes 3 and 4. So let's talk about some of the differences between the two approaches here. So when you're using an endogenous protein, like a housekeeping protein, it's normal to use something like GAPDH, tubulin, or actin. You could potentially have a limited detection range there. You are just getting a single protein signal, so it's just that one protein you're relying on. Now, the cellular treatment, how you're treating the cells with its growth conditions, the drug treatment, the disease state, maybe a different isoform, that could all lead to varying expressions of that supposedly housekeeping protein. So you'd really need to validate that it is not changing under those different treatments. You will still obviously need to optimize your antibody in terms of the dilution on being able to recognize that protein target. But it is a very well known and widely used method, which a lot of people use to try and help to overcome like these problems with loading and transfer efficiency, as we said. But another approach you could take is to do a normalization against total protein. So we could have something like a Cy5 pre-labeled protein. There are different detection strategies to go in there to do the total protein. The advantage here is you get a very broad detection range. So you're seeing all of the proteins hopefully within the sample. You're going to sum up all of the protein signals within that lane to do the normalization. And then this is going to be minimally affected by the different treatments or growth conditions or drug, disease states, whatever, because you are going to look at all the proteins that you know you've loaded a certain amount into each lane. So maybe it's 20 micrograms in each lane. So whether proteins are up and down regulated in there, it doesn't really matter because your total amount of protein is going to stay the same. It's antibody independent because obviously you're just pre-labeling the protein in there, and it is definitely a more current method that people are looking at because it's a lot more robust. And you can see now a lot of the journals are saying to maybe avoid housekeeping proteins and then to kind of use this approach with total protein normalization. So you've got journals such as the Journal of Biological Chemistry, American Journal of Physiology, and PLOS ONE as well. So it's definitely becoming more of the go-to methodology to kind of negate all of these potential issues we could have. So if we look at this situation here, we're doing lane-to-lane normalization with total protein. So you can see we have these different lanes, different proteins being run in each channel. And then we're going to basically to do the normalization, we'll take that protein of interest, band A, which is the antibody is recognized, and then all of the other proteins, including potentially that protein that you're interested in, is going to make up the total protein. And then you can see there on the upper panel, we have no normalization on the left. So it's given one story, but then we do the in-lane normalization. You can see actually the amount of protein there is very similar in lanes 1 and 2, which we'd have missed if we hadn't have done the normalization technique. So the labeling we just mentioned there using this Cy5 label, basically you're going to use an NHS ester reactive group to bind the protein to the dye, or the dye is going to bind to the protein. And then you obviously need to have the correct pH. So there'll be a labeling buffer to have that between 8 and 9. You can see that there. And then we're going to use this quick stain Cy5 to start the reaction and then to terminate the reaction. And that reaction could take 5 minutes in a quick protocol or a longer one. You could take up to 30 minutes to do the reaction, depending on the end goal of your study. And then to terminate the reaction, you're just going to add the loading buffer with dithiazolidine in there, heat the sample, and that will stop the labeling reaction. Now, when we use this kind of approach, we have very good sensitivity. So we can get down to about 0.2 nanograms, 200 picograms per band. The linearity is, 10 to the 3 orders there. It's very robust in terms of the concentration of sample that we're using. So you can see that there from 20 micrograms per microliter up to a nanogram per microliter. You get very sharp bands. You can see that on the right-hand panel there. So we're seeing the linear dynamic range on the left, but then on the right, the protein is present, but you might need to adjust the contrast on the screen to see it. But the raw data is really going to have all of that information that you perform the analysis on. And then there's a very important journal here, Analytical Biochemistry, that has a reference talking about how robust this methodology is. And we're going to look at some of that in more detail now. So in this experiment, they basically did different loadings of CHO cells. And then they looked at different ways to do the normalization using different housekeeping proteins of the total protein. So in this case, they looked at using GAPDH as the housekeeping protein to do the normalization. And you can see there we have a very big coefficient of variance, 53%. So obviously GAPDH is not a good choice as our housekeeping protein. But then they tried with tubulin. So with tubulin, again, same experiment, different protein as a housekeeping protein. You can see now the coefficient of variance is only 18%. So that's a lot tighter. That's a lot better. But then in the final example here, you can see that they used the total protein. So that's the Cy5 there. The target protein is with Cy3. And then when you do that normalization against the total protein, you can see the coefficient of variation is down to 7%. So that is really, really good, tight data. And then you can use this approach, total protein normalization, just using the fluorescent workflow. So you'd have Cy5 maybe for the total protein, and then you could have a Cy3 labeled secondary antibody. If you had a near-infrared system where you only had like the 2 near-infrared channels, that Cy5 label will actually crosstalk into that short infrared. So then you could still use that same protocol with that same reagent. And if you really want to get really great sensitivity and you're using a chemiluminescent workflow, then you could still use the Cy5 for the total protein normalization as long as your camera system had the ability to do fluorescent as well as chemiluminescent detection. And here's an interesting example where we've got a 2D gel. So the total protein in here is with Cy2, so you can see that colored in blue. And then we have our protein of interest, GSK, it's colored in green. And then you can see the phosphorylated form, which has been colored red. So that would be a Cy5 conjugated secondary antibody against the phosphorylated protein. Now, to do that normalization approach, either with the housekeeping protein or with the total protein, we're going to use a software package. You could use whatever software package and then extract the volumes and put that into Excel and do the division in there. But actually in this software, the ImageQuant Total Lab version 11, it's built into the software to be able to do that normalization procedure. And then here we're going to use this gel, and blot analysis module. So the first step in the process is to define the lanes. So we can see in the example here that the lanes have been defined, and you can see there's obviously some loading issues with this particular example here. And then you want to remove the background. So there's different ways to remove the background in the software, but here we use the rolling ball method, and you really want to have base-to-base resolution of those peaks, which we have achieved here. Then you want to detect the bands. Again, as an automatic detect in the system, or you can manually detect the bands or change your parameters to get it tighter, but it usually works pretty well here. So you can see all of our bands have been detected. And here you have two options with the to do the total protein. You can either draw a band over the entire lane, or you can just say, I want to use the total lane volume. Pretty much it's going to be the same in both cases if you pull the band to cover the whole lane. But it might be that when you drew the lanes, there might be a little artifact at the top or a bit of dye at the bottom and the dye front that you might want to avoid. So then you could do, like I said, you can draw a band within the lane, which covers everything of interest without any kind of artifacts. And then this is a summary here showing that you have, you can see the purple dot. So one corresponds to the housekeeping protein and the other to the total protein. And you can see from the drop down here, you could normalize against the selected band, the largest band. You could do the housekeeping protein, or you can see that last choice, which we're going to do here, is do the total protein. And when we do the normalization, so you can see on the right-hand panel, the green line that's going up and down here, that corresponds to, the poor loading with different loading amounts. But then you normalize and everything should be identical. And then on the left-hand panel, again, the green line is showing the protein of interest, how that's going up and down. But then when we normalize, you can see it makes it a bit smoother. And that is like the true data there when we've done that procedure. So there's just some resources here. So if you want to work with the software, there's a 21 free trial license you can download from the website. And then there's some information on different imaging systems, also about the protein labeling kit we mentioned there, and just the dyes in general if you want to kind of take a different approach there. So thank you very much for your attention, and thank you.
This episode is kindly brought to you by Cytiva. Cytiva is a trusted expert in protein research, supporting scientists across the Western blot and imaging workflow. Their integrated portfolio spans high-performance imaging systems, Western blot reagents, membranes, ECL, and consumables, designed to deliver sensitive detection, reproducible results, and streamlined workflows. Together, these solutions help labs generate reliable protein data with confidence, from sample to image. To learn more, check out the links in the description. See you next time.